Hello everybody, welcome back to our channel, Biology again. And we are glad we are going to B3. And we will start on 3.1 gas exchange. But what is B3? B3 is the form and function of organism, okay, to the whole thing. Today we talk about what's form and function, talk about respiration, okay. Gas exchange, okay. We talk about gas exchange in human, using the lung, and in the liver, okay. Two things, okay. So gas exchange is in general is an essential vital function of organism because we have respiration, right? So we have to take in oxygen and we have to release carbon dioxide to the surrounding. Of course, we have learned that surface area to volume ratio decreases as the size increases. So for the whole organism, wow, the size is so big. So this ratio is very low, not very efficient. On the other hand, when we get bigger, okay, even the distance from the center of the organism to the exterior increases too, okay. So there's a big separation. And then this is the what happened in the lung. Okay, there are three parts. Okay, the first part is the how the oxygen and carbon dioxide are released or absorbed from the surrounding, and then how it is transported through the body. Okay, first of all, the pulmonary ventilation is the breathing of the lungs. Okay, pulmonary ventilation consists of two processes: breathing in of the air, right, and breathing out of the air. In pulmonary ventilation, oxygen moves us into the alveoli, and carbon dioxide moves us out from the alveoli. So alveoli is the way the air, oxygen go in and carbon dioxide go out. After going inside the body, okay, gas exchange happens. Okay, it's the alveoli, and the gas exchange happen between the alveoli and the capillary. So the oxygen and carbon dioxide transported to the red blood cell. Okay, and the red blood cell transfer the gas, okay, to the whole body. Okay, by ventilation, they will have a high concentration of oxygen and a low concentration of carbon dioxide. Okay, than the capillaries. This concentration gradient mixes oxygen to diffuse from the alveoli to the capillary, and on the contrary, from the capillary back to the alveoli for the carbon dioxide. So it's a natural diffusion due to concentration difference, not an active transport. No energy is needed. Okay, just breathe in the air and bring out the air. Okay, so the bloodstream must transfer the oxygen from the capillary to the body cell for cell respiration, and then the stream also transfer the carbon dioxide released in the cell to the capillaries. And property of the gas exchange surface. Okay, so the gas exchange surface must be permeable to oxygen and carbon dioxide. Right? But oxygen and carbon dioxide only permeable to the cell membrane through when it is dissolved in water, not in the gas state. So the gas exchange surface have to kept moist. Okay. So increase the efficiency of gas exchange. You remember the gas exchange surface have to be thin and have a large surface area. And it is gas exchange is a good thing, no need for energy. It is a diffusion nature. Okay. When you breathe in the air it has a more oxygen content, right? So this high oxygen content diffuses through the alveoli to the bloodstream. Okay. When the bloodstream bring in the carbon dioxide, okay, the carbon dioxide content of the bloodstream is more, okay, higher. So the carbon dioxide diffuses to the alveoli naturally. So it is good. It is a gas exchange. It's a diffusion in nature. Okay. So what we need to do is to maintain the low oxygen concentration and the high carbon dioxide concentration in the body cell. Okay. And also to maintain a high oxygen concentration and low carbon dioxide concentration in the environment. Just keep them moving around, keep the air moving around, keep the bloodstream moving around, and everything will do well by diffusion. And how do we do it? Okay, of course, we need to learn a little bit about the structure of the lung. Of course, you know, everyone know it, okay, in the basic biology, okay. Very next, okay, trachea and then the bronchioles, okay, and now from the alveoli, okay, and then all are smaller and smaller branching out, right? So when this branching out happens, okay, this surface area increases, and at last, going to alveoli, it's a bunch of alveoli there. In the old time, we call it junior form, called air cell, right? Now the alveoli, okay. And also in the alveoli, we need to keep it moist, right? And then the thing keep it moist, we call it surfactant, okay. Surfactant keep it moist. Okay. And this is important to keep it moist. Okay, if it's not moist because there's a little bit acid, not alveoli. Okay, if they're moist, they will collapse with each other. Okay, and the efficiency will go down quickly. And then adaptation of alveoli we have talked about. Okay, and in IBDP we need to learn that there are two types of hemocytes, two types of cells inside the alveoli. It's okay. First of all, you need to lower. Basically, the alveoli has a spear shape. Okay, to maximize the surface area. Okay, and also it is very thin. Okay, a few cell thin. Okay, so that the carbon dioxide and oxygen can diffuse through it to the capillary easily and quickly. And also you need to remember, okay, who will squeeze the surfactant and who will transfer the oxygen and carbon dioxide. Very easy. The type 1 is for the transfer of oxygen and carbon dioxide for gas exchange. Okay. But the type 2 lymphocyte is for the secretion of the surfactant to keep it moist, to keep them not collapse. Okay. And after the oxygen going inside, okay, or the oxygen is bind to the hemoglobin in the red blood cell. Okay, the red blood cell has hemoglobin, and this heme okay bring the oxygen out. Okay. Also this heme also brings the carbon dioxide into. We talk about the interaction between this binding of the oxygen and carbon dioxide with the hemes. Okay, later in HHL. Okay. So remember type 1 and type 2. Okay, type 1 lymphocyte is for the transfer of gas. Okay. Type 2 lymphocyte is for the secretion of surfactant. Okay. And the surfactant has many different effects. Okay. Reduce the surface tension and make the water pass through easily and prevent the collapse of the alveolus. Okay. And if they stick together and then they can't work properly. Okay. And then you have to have a drawback, remember, for premature babies, okay, born earlier than 30 weeks, okay. The type 2 lymphocytes, okay, always are not fully developed, so lack of surfactants. So the lung has a high surface tension, and then the alveoli will stick together, so they can't breathe properly. They have to wait for a few weeks, okay. It's the most reason, okay. And also alveoli is a way where it's contact with the surrounding, right? The outside here, so they also have some microfibers there to defend against the foreign substance. Okay. And for the lung, okay, first we know how to keep it healthy is important. So we learn what's the disease. We call it COP, COPD. Okay, chronic obstructive pulmonary disease. There are not some kinds of COP in the world. We need to learn too. Okay, in IBDP. And how to detect whether you have this problem or not. It's easy whether you have a forced expiratory volume is high or low. If you can breathe in well and breathe out well, okay, the maximum volume of air that can be expired in 1 second. You can do it well, then you don't have the problem. And if you can't do it well, it means that the function of the lung is going down and down. And there are 3 categories: mild, moderate, or severe about the problems. Okay. And there are 2 kinds of disease we need to learn. Always happen in IBDP. Okay. Emphysema is one. Okay, this emphysema, this is a disease that will destroy the alveoli. Okay, so we feel alveoli breathing becomes difficult. Okay, and emphysema is caused by air pollution, coal dust, and fumes. Okay, and the other thing is we learn to learn elastin. Elastin is another thing. Okay, that keep the elasticity of the lung. Okay, so you can breathe in and breathe out quickly. And then with the elastin that degraded, the lung loses elasticity and breathing becomes difficult. So the degradation of elastin is also caused. Okay. So fewer alveolar and degradation of the elastin is caused. And the other kind is more serious, we call it lung cancer. Okay. Lung cancer is caused by the mutagen, or carcinogen. Okay. And then lung cancer may grow to block the bronchus or trachea, and then the patient will experience shortage of breath. Also, the lung cancer may cause the accumulation of fluid around the lung. Okay, so the fluid there, so patient cannot fully expand the lung during inspiration. Okay, so all this is reflected by a low inhalation volume. Okay, so the mortality rate of lung cancer is high. Okay, we know our smoking has a positive correlation between this thing. Okay. And nicotine addiction causes people to continue to smoke. So if people doesn't want doesn't want to smoke anymore, they may go through a nicotine replacement therapy to quit smoking. And what's the pulmonary ventilation mechanism? Very simple. Okay. If the lung expand, volume decrease, and the inside pressure decreases, right? And then the the atmosphere has a high pressure and they push the air in. So how to push the air out? So we contract our thoracic cavity. Okay, contract, volume decrease and pressure increase, and with high pressure we push out the air. But how to do it? The mechanism is like this. It's by the lung, two ways. Okay, two ways. Okay. Well, when you want to inhalation, okay, remember inhalation you need to increase the volume, so the rib cage move up and out. Okay, and the diaphragm move down and flat. Okay, and the volume of the lung increases and the pressure decreases, and we throw in naturally. How to push it out? It is exaggerated. Okay, and then the rib cage move down and in. Okay, and then the diaphragm move up and down. Okay, become a doom, so the whole lung volume decreases, pressure increases, and the air will go out. Okay, this is exaggerated. Actually, not that different. And look at the two kinds of muscle. Okay, we have to remember in the rib cage there are two kinds of muscle. Okay, external is outside, internal is inside. Okay, there's external intercostal muscle and the internal one. And just when you draw it out, you can just observe whether they are longer or shorter. Okay, you can see when it is moving up and out, the external one is shorter, internal one is longer. On the contrary, when with moving down and in, the external one is longer and the internal one is shorter. So what do you mean by longer is relaxed, right? Shorter is contract. So when it breathing in, the external one has to contract, internal one has to relax. On the contrary, when we breathe out, okay, external one has to relax and the internal one has to contract. Similar to the diaphragm, okay, when it moves down, okay, it is shorter. And we move up, it's longer. So which one is contract, which one is relaxed, okay? So when the inhalation is contract, this is relaxed, okay? That's the concept. So all the thing is here, contract, relax, okay? This contract and relax is hard to remember about the internal intercostal muscle. You need to draw it out, okay? Then you recognize, okay, longer is relax, shorter is contract, okay? And then under hypoxia, okay, when you are lack of oxygen, okay, and then your muscle will contract and relax quicker, okay, to have more ventilation, okay. And volume of the lung, how to measure the volume of the capacity is via water displacement method. Remember, you blow inside, okay, see how much water is displayed, and that is the volume of your lung. Okay, there are two kinds of measurement. One is the vital volume, just the maximum, how much you can breathe in and breathe out, call it vital volume. And the other is what's the reserve for you for the normal breathing and the maximum breathing? What's the difference between that? That is the vital part, right? When you have a big exercise, okay, you that part is essential for you. So we call it inspiratory reserve. Okay, when you breathe in normally, what's the volume? And breathe in heartily, hardest one, okay, strongest one, how much you can. And then the volume, we call it inspiratory reserve. Expiratory is also okay. When you exhale the air, okay, normally how much volume, and then make it hardest, how much volume, and the difference is the reserve. Okay, the reserve is the buffer for different scenario. Okay, and how the monitoring in the mild and vigorous exercise, when you do exercise, especially with your athletics, okay, they are not monitoring to you, okay. What about the lungs property? We measure the oxygen concentration, ventilation rates, and total volume of ventilation. All this things go up and down together. When you do exercise, all the thing goes up. When you stop doing exercise, all the thing goes down. But there are two kinds of exercise. When it is mild exercise, no problem, okay, aerobic respiration, right? So it goes up, and then when you stop exercise, it goes down quickly. But you have vigorous exercise, what happened? Remember, not enough oxygen, so anaerobic respiration happens, and lactic acid appears in your muscle, okay. And even after you do the exercise, you need to have a high oxidation consumption, okay, to oxidize the lactic acid, okay. So it drops slowly after a vigorous exercise, majorly because you have an anaerobic respiration, lactic acid there, and you take more time to oxidize the lactic acid. And then about the leaf, okay, the gas exchange in the leaf. So we learn the leaf and the lung. We learn one already. After the leaf is the same simple. Okay, remember at top, at the bottom, okay, at the upper surface, okay, it has a waxy cuticle because they don't want so much respiration. Okay, because so much expiration due to the sunlight, okay, directionally. And on the back, on the lower surface, it's okay, okay. They allow a little bit transpiration, okay. So they have a gas cell there, stormer there, and the stormer is for the gas exchange, oxygen go in and go out, hydrogen and then carbon dioxide and water, okay. And it is the open and close of the stroma is something called storm, okay. It's controlled by the gas cell, okay. And the waxy cuticle protect the liver, but only at the upper surface because there's a low permeability, reduce the evaporation of water, and also it stop the gas exchange. So the lower epidermis does not have a waxy cuticle, okay. So it can have a gas exchange, okay. So it has a stroma for gas exchange, okay. And water also evaporates, okay. So transpiration and gas exchange happen both at the lower surface, okay, at the storm. And how to control the open and close of the stroma. When you draw these two diagrams, you remember when the gas cell is turgid, okay, full of water, okay, it open. Yes, that's good, okay. When it have water, so it doesn't mind about transpiration, right? When it doesn't have enough water, so it become flaccid, and then become flaccid, the stroma close, okay, decrease the transpiration. That's a good mechanism, okay. And how can we detect whether there's gas exchange? Okay, we can detect it by water, right? How to detect water? By copper chloride paper, right? With water, it will turn pink. Okay, so put the blue copper chloride paper on the back of the leaf, okay, to see how many pink spots after some time, and then you know how many stroma are there, approximately, relatively. Okay. Of course, later on you can use a microscope to directly observe the number and count the number of stroma there. We can do it. We have high resolution microscope to do so. Okay. And the transverse section, okay, cross section, okay, vertical cross section of the dicotyledonous leaves, okay. It's like this, okay. It has the, you can see that the stroma is at the bottom and the gas cell is there, and then when the gas move in and move out, okay, there is a air space, okay, for the gas move in and move out. Oxygen in and carbon dioxide out, okay, and then spongy be survive. And the air will move through the xylem and the phylum and all the cell, okay. And also remember the wing, okay, the xylem and the phylum will support the leaf too from collapsing. Okay. So what's the abiotic factor affecting transpiration? Okay. You know, transpiration is evaporation of water. It is a consequence of gas exchange. So when the transpiration is high, gas exchange is also high. Okay. So what's the factor affecting transpiration? Of course, outside has to be dry, right? We want it to be large transpiration. Sunlight has to be strong, right? And the wind has to blow away, right? And all this thing is the reverse, and then the transpiration will be weak. So in humid surrounding, transpiration is weak, right? Low sunlight, transpiration is weak. Low wind, low airflow, and then the evaporated water will be accumulated, okay? And the evaporation transpiration will be low, okay? So easy to think about it. So remember, when transpiration is high and low, okay, gas exchange is high and low at the same time. So there's some laboratory skill to measure stomata density, okay. Of course, you can use the copper chloride paper, okay, observe the pink spot, or you can use directly use some high power microscope, okay, to count the number of stoma. But of course, you need to repeat the count it, okay, at different area regions, okay, to make sure you have the correct unbiased data. And then we go to additional high level. Remember what's additional high level? Adaptation. Yes, adaptation. Okay. First of all, is the adaptation for the transport of oxygen. For example, for our lung. Okay. How about the hemoglobin? Okay. What do the hemoglobin do? Okay, is about the binding of oxygen. Okay. So hemoglobin has to transfer the oxygen from the lung to the other part of the body, right? So when it has a higher oxygen content surrounding, the hemoglobin has to bind to more as much oxygen as possible, right? So it's dropped, okay. So when the hemoglobin goes to the body parts, okay, where the surrounding oxygen level is low, it has to release the oxygen out, right? So it has to have a low affinity. So the affinity of oxygen to the hemoglobin has to change, okay, by the surrounding oxygen level, okay. So the concept is called cooperative binding, okay. when the oxygen level is higher surrounding it, okay, oxygen will bind to the heme, and the heme can bind more oxygen, okay. So the affinity of oxygen will be high, okay, when the oxygen surrounding oxygen content is high. It is for what happened in the alveoli, okay. And then we call it allosteric binding of carbon dioxide in the hemoglobin. Carbon dioxide also bind to the heme too. Okay, carbon dioxide is also bind to the hemoglobin. Okay, when the concentration of carbon dioxide is high, the concentration of oxygen is low. Okay, so more carbon dioxide is bound to the hemoglobin. Okay, to enhance the effectiveness of the release of the carbon dioxide from the body cell to the red cell to the hemoglobin. Okay. And what happened to the young babies, okay, the photo hemoglobin? The photo hemoglobin has a different composition than the adult hemoglobin. Okay, they need to get more oxygen. Okay, so the photo hemoglobin binds oxygen more strongly. Okay, so they can get more oxygen from the mother's bloodstream. Okay, and we have to learn effect called Bohr shift to explain the thing. Okay, it is a concept. When the concentration of carbon dioxide increases, okay, carbon dioxide is a little bit acidic, right? Carbonic acid, right? So the blood pH decrease, become acidic, and then the affinity for oxygen will decrease. Okay. Why is this important? It's a Bohr shape is important to explain the S curve at the back. Okay. So it is the S curve of the oxygen dissociation curve. Okay, is one hand is the surroundings oxygen concentration, the other is amount of oxygen bound to the hemoglobin. Okay, this is important, right? So when the oxygen concentration surrounding is high, affinity has to high. Okay, so the hemoglobin absorb as much oxygen as possible. When they move to the body, and then the surrounding oxygen level is low, they need to release the oxygen they have, right? So the affinity of the oxygen has to be low. So this S shape is essential. And what do the Bohr shape explain? Okay, this part or that part? Yes, it's about this part. Okay, when the oxygen concentration decreases, okay, relatively the carbon dioxide concentration increases, right? So about the broad shape, okay, fewer oxygen molecule are bounded to the hemoglobin, so we release it. Okay, this one is by the cooperative binding of oxygen. Okay, so these two parts have different explanation. Remember, this part is broad shape, and this part is the cooperative binding of oxygen with hemoglobin. So remember how to explain the thing. And that's all about the gas exchange. Okay, we learned it like SL and HL. In SL, we talk about the lung and also talk about the leaf. Okay, in the HL, we mostly talk about the lung. Okay, the adaptation of the lung for gas exchange. Okay, so you learn part of it. Okay, it's about the organism. Okay, so see you for next biology day. Okay, if you like our channel, remember subscribe, like, and share with your friends. Okay, see you next week. Okay, bye.